In this section, I'm going to talk about the questions in BB350's problem set D. The first question asks, what two parts of an amino acid are joined together in making a peptide bond? And this is pretty straightforward. If we look at how peptide bonds are oriented inside of cells. If we think of an amino acid as looking something like this, we could see an R group, we could see an H, we could see a carboxyl, and we could see an OH. We could also see over here an NH3 plus, something like that. So this is one amino acid, and this amino acid might be at the end of a protein. That is, it might be the very first amino acid that makes it into a protein. The next amino acid that makes it into the protein would have a structure that looks like this. Yep. I can't write today. OH, H dash NH3, like so, okay? And actually, I'll leave that as it is. Well, in making a peptide bond, what happens is that the carboxyl of one amino acid is joined to the amine group of another amino acid, and I should have written these as just 2's for right now and leave that as a 0. Okay, so I've got a 2 here. What I'm going to do then is I'm going to pull the proton off of this guy, and I'm going to pull one of the protons off of this guy, and I'm going to pull the oxygen. Well, so I've lost an OH, and I've lost an H. These two guys go together to make water, and these two guys end up being joined to each other. So we've made, at this point, a peptide bond that has joined the alpha carboxyl of one amino acid with the alpha amine of another amino acid, splitting out water in the process, as we can see here. Now, I leave this on the screen because it also helps to answer the second question, which is what parts of a polypeptide backbone behave as if they were a double bond? And it turns out that the part of the polypeptide backbone that behaves as if it were a double bond is actually the peptide bond. The peptide bond has qualities of arising from electronic resonance that make it behave part of the time as if it were two bonds instead of one bond. And when that happens, this double bond can't rotate. A single bond can rotate, a double bond is rigid, and this guy behaves, most of the time as if it is a rigid double bond. The last thing I'll talk about, or answer the question of, is name the forces stabilizing alpha helices and beta strands. So let's take a quick look at that while I'm erasing this from the screen, and I will tell you a little bit about that in the process. Okay, so let's consider what we're thinking of here is secondary structure. Secondary structure of proteins relates to regular repeating structures that we see over and over. In the case of alpha helices and beta strands, we see something that might look like this. And I'm not a very good artist, so excuse me for that. So let us consider the alpha helix over here, the beta strands that we have over here. We remember that secondary structure arises as a result of interactions between amino acids that are close in primary sequence, meaning close together on this chain. If we look at these forces that are actually stabilizing this, this is in the form of hydrogen bonds that I'm going to draw as little dotted structures here and little dotted structures here. Okay? So hydrogen bonds actually help to stabilize this structure. Okay? Similarly over here, if we look at the bonds between individual beta strands like we see there, we see that these guys are oriented so that they also have hydrogen bonds that help stabilize their overall structure.